Okay, welcome back. Today we're going to cover Chapter 3 on Mendelian genetics. So some of the goals of this chapter are included here. By the end of this chapter, you should be able to define and distinguish between a character, trait, and a phenotype. So these are three different things that we use to describe how an organism looks. And then a gene, allele, and genotype. These are three different things that we describe the units of inheritance for. So, what controls the phenotype, what is within your DNA that is able to influence how an organism looks, for example. And then this is the first chapter where we're going to discuss how we use analysis of both genotypes and phenotypes of individuals in order to infer kind of the inheritance pattern and how inheritance work. So typically what we do is we look at phenotypes and genotypes of individuals, and then we perform some sort of cross and look at the ratios that we observe in those offspring. And so that's referred to as genetic analysis, and it's a very important part of the first third of the class. This will be kind of come up over and over again. So we're starting very simply in conditions where the rules are easy, and then we're going to make things a lot harder as we go through the following chapters. Understand the difference between a natural and an artificial population. In some situations we control the breeding, in some situations we don't. And that's kind of the essential nature of the difference. Describe how geneticists use artificial populations for genetic analysis. How do we use intercrosses? How do you use test crosses? Integrate probability rules to the analysis of simple genetic traits. Probability comes into play because there are multiple outcomes, and so how do you use probability in order to calculate the proportion that you expect to see for a given trait. Apply the chi-square test to genetic crosses. Students always hate this part. Unfortunately, it's a necessary part of genetics that you have to know. And then finally, differentiate the genetic basis of traits and genotypes of individuals by analyzing pedigrees. So this is an example of natural populations. This is important for aspects of human health. If you're interested in becoming a doctor, this is probably something that you'll be very, very interested in. So this chapter covers the work of Gregor Mendel, who's considered the father of genetics. Prior to Mendel, many believed in blended inheritance, the theory that offspring receive a mixture of characteristics from their parents, resulting in offspring with intermediate phenotypes. And he kind of turned this theory on its head. And he did that because he did a lot of very careful experiments using pea plants. So Mendel instead proposed particulate inheritance, the theory that characters are determined by discrete units that are inherited intact through generations. And these are genes, and the important part of this is that we each have multiple alleles of each gene. And so if we integrate this with the previous chapter, we know there are two homologous chromosomes that are present in every single cell. One that we receive from the mother, one we receive from our father, and that means that we also have two copies of each gene. And because there can be variation between these homologous chromosomes, we actually have two different genes that can have different versions. So Mendel primarily studied pea plants. He studied pea plants that had variable phenotypes that we will discuss in a little bit, but by following the inheritance of those characters, Mendel proposed a number of different rules. So first, characters are unitary, that is, they're discrete. So characters are purple versus white, tall versus dwarf. So we're not looking at the continuum. We're looking at phenotypes. We're looking at characters that are kind of binary in nature, or at least discrete in nature. This can be distinguished from things like human height, which there's a quantitative nature to that phenotype. Genetic characteristics have alternate forms, each inherited from one or two parents. So today we call these alleles. So you receive a version of each gene from your mother and father, and those genes can be different, those versions can be different, and if they are different, then they actually have different alleles. And so he called these forms, we call these alleles. One allele is dominant over the other. So when you have two alleles, because your mother and father gave you different alleles, one form is always dominant over the other allele, and the phenotype reflects what that dominant allele is. Inheritance. So when you look at what offspring you are going to have, you have two versions of each gene, and so you need to produce gametes that have only a single copy of each gene. And so gametes are created by random segregation, and it's pretty much completely random. So if you have two alleles, each gamete has an equal likelihood of having one of those two alleles. There's not one that has a preference over another. And finally, if you look at different traits and different characters, they have independent assortment. In modern terms, genes are unlinked, but one way you might think about this is that the genes are present on different chromosomes, and so they sort completely independently. So these are very simple rules. We know there are many exceptions to these rules, so this chapter is kind of a subset of all possibilities, and we're going to expand upon these in Chapter 4 and 5. Where these rules end up being violated. So this chapter is broken up into 4 parts: analysis of a single character in artificial populations. Part 2 will be the analysis of a single character in natural populations. Part 3 will be analysis of multiple characters at the same time, and then finally Part 4 we will cover chi-square tests.